I will be presenting on the effects of cholesterol on antimicrobial peptides, and this is something I've been working on for the last few months. So before we begin, why bother? Why does my research matter? So a lot of people before, such as my mentor Kevin, they have worked on creating certain models of how peptides work regarding certain characteristics depending on adding certain elements such as the membrane, the copies of the different ratios of POPC and stuff like that. But no one actually has done a research how cholesterol could affect antimicrobial peptides. And this is something that could be interesting and really exciting for other previous and future work in the field, because perhaps cholesterol might have an effect on how antimicrobial peptides could work. And so yeah, I'll talk more about how and show you a picture of how this actually looks like and affects the membrane. And before I talk more about what I'm doing, let's talk more about antimicrobial peptides. So what are they? They are a host defense of peptides. So peptides are basically from 2 to 50 amino acids that come together to achieve a goal. In this case, they attack bacteria. And what is so special about AMPs is that, or antimicrobial peptides, is that there's been a huge interest by drug makers in order to find certain cures for certain diseases, such as skin diseases, lung diseases, and some people even say cancer. And we can see here that we have 4 different types. And they're all very interesting, and they're all very interesting in the sense that there's a lot more to be discovered in research. In my case, I'm specifically researching meganing 2. And something notable about meganing is that it has an alpha helix shape. So now let's talk more about how do they work. So like I said, antimicrobial peptides are a mechanism of defense. And as we can see here, how they work is that they disrupt the membrane. How? A couple of things. The first thing is by hydrophobic interactions when it's weak, and also when it's very strong, it is by hydrophobic interactions and also electrostatic interaction. And so yeah, so we can see that we have bacterial and plants and mammal membranes from different cells, such as prokaryotic and eukaryotic. And we can see and think about how we should expect a different result for the weak interactions when it has cholesterol. And again, also why adding cholesterol could have some interesting results is that plants and mammals membranes and they have, such humans and animals, they have cholesterol, which could improve our understanding of future drug making. So going back to my research question, so how does adding cholesterol to a computer model change the results? What do you expect to see, and why is it exciting to do research like this? So the proposed hypothesis is, like I said, how does cholesterol affect our model? So in this image right here, the first one is actually from my mentor's Kevin's previous work. So what he did is that he did a similar work, but he didn't use cholesterol. So we can see here we have 2 membranes. We have an anaiotic composed of POPC plus POPS, and we also have a POPC neutral. And what we can see here is that both membranes show a strong interaction, and we should expect something similar when we add cholesterol because of the different charges of the membranes. So we should expect to see the peptide not as close to the membrane due to different charges. And then for the proposed evaluation, I plan to simulate, so for the proposed evaluation, I plan to simulate 20 nanoseconds for 20 nanosecond simulations for 5 copies in my gaming system, composed of 50% POPC plus 50% cholesterol. And here, this image is something, it's a model I created using Charm GUI. And this one specifically has 50% POPC and 50% cholesterol. So this one, I think, is one of the copies. I think it's the 4th one that I selected. So how do I plan to carry this out and make this a reality? The first thing is that I'm using an HMM model. And like I mentioned before, I plan to use Charm GUI to make the different copies and create the system. Then I plan to use NAMD to run the simulations. And lastly, for the analysis, I plan to use clusters, specifically LUPT, to run simulations and do the analysis. And then conclusion and future steps. Like I said, this is something that has never been done before. It's a very novel work. And it is so important because first, it has implications in our understanding of plants and mammals' membranes, which again is really important because it could be used to enhance our understanding of future drugs that could be made. And number two, this could have a significant impact on previous work that has been published and of course future work. And as I look forward to the future, something that I think is very important, as I look forward to the future, I will be presenting this work in the undergraduate conference happening in a month or so. But beyond that, I plan to keep going and finish this work and aim to publish this work and keep going with the bigger picture, which is to understand more about antimicrobial peptides. And yeah, thank you very much.